I'll be talking about abiogenesis tonight. And that's one of those funny words that you can figure out what it means if you take it apart. Currently, it's the most prevalent scientific explanation for the origin of life. And when you take this word apart, means not, bio means life or living, and genesis is origin, beginning or origin of life. So this word is really telling us this is the non-biological origin of life. And that's the current scientific explanation of how life arose, that there was this non-biological process. The origin of life is one of those big questions in life. It tests our faith, taxes our science, it challenges the deep thinkers, it defines our identity, and is an integral part of our worldview. You can imagine that depending on how you imagine where life came from, you're going to have different perspectives on what the human race is all about. If we're just highly evolved pond scum, you're going to have one view. If you see yourself as a child of God, you're going to have a totally different view. And so this is one of those questions that is almost essential to civilization to get right. And all civilizations, one way or another, try and develop answers to the question of where life came from. Tonight we're looking at the scientific aspects of this question, so we want to get a definition of science that we all sort of agree upon so we'll have some common ground. One definition of science is that it's a search for natural explanations of the world around us. Another one is that it's the reasoned effort to rid the world of superstition and religious myths. Or it's a method for testing about the physical world through observation, analysis, and experimentation. Or it's all of the above. I won't have a quiz, but definition C is the one that I think is the best definition of science. Definition A is something that often happens. We do get natural explanations about the world around us through the process of testing and observation and experimentation. But it's not limited to just that. If you were an environmental chemist and you're trying to look at the distribution of lead in the ocean, you would be making decisions about whether the concentrations that you saw were totally natural, whether they were pollution, which would be anthropogenic, man-derived, or some combination. So you're actually discerning the difference between natural and other processes. Definition B is sort of a red herring. I just threw that in to see if people were strongly biased against superstition and religious myths. And of course, if either A or B or C is not there, then it can't be D. But method C really is all about how science is done. It is a method for testing our ideas. And we compare them against the physical world. We want something that is unbiased, that doesn't have philosophical overturns. We want something that we can use as a yardstick. Use as a yardstick for comparison. Science is also not about believing. If science is about observation and experimentation, then we really know what happens when we have a certain set of conditions. You're not extrapolating. You're not imagining what happens between things. You know. Belief, on the other hand, requires that we have faith in things unseen or trust in things hoped for. That's an entirely different set of standards. It doesn't mean that belief is wrong and that science is always right, but they're two different things. And clearly, science works to this different standard where we test our ideas versus reality. So why am I, as a chemist, interested in or speaking to you tonight about the origin of life? Well, if abiogenesis is about the origin of life, shouldn't we be listening to a biologist? Well, before life begins, there's only chemistry and maybe some physics, but there's no biology. So you really do need a chemist to look at this issue. Chemistry is also a repeatable thing. Chemicals don't evolve. If you have a set of chemicals now, they're the same as they were 1,000,000 years ago, and they'll be the same as they will be in 1,000,000 years in the future. They'll do the same thing all the time. This makes chemistry testable, so we can really test the ideas we have about how life originated. Some of the ideas that people are considering and testing is that life is the result of unguided chemical reactions and random processes. Another idea is life is the result of purposeful physical laws and initial conditions. In other words, somebody stacked the deck and set things up for life to happen. A third idea is that the laws of nature and the fine-tuning of the initial conditions are insufficient to account for the origin of life. Something has to happen continually along the way. So you just can't stack the deck the way you actually have to cheat as you go along. Or limiting our options to natural processes alone prohibits the origin of life. These are all ideas we can test through experimentation. One of the most famous experiments about the origin of life is the Maillard experiment. In 1953, Stanley Miller, he really stunned the world with the results of his experiment, where he announced that amino acids could be made from a mixture of reducing gases with an electric spark. This totally caught the world by surprise. And subsequent to his initial analysis, people have identified in that mixture all the sugars, all the nucleic bases, a lot of the fatty acids, the things that you need as basic building blocks for assembling a cell. Virtually everything you want to find was in that mixture. Now, the news stories describing this work, they appeared on the front page of newspapers worldwide. Big banner headlines, life in a test tube. This really caught the attention of the world. And the experiment, in most cases, was hailed as the first step in the origin of life by a purely naturalistic pathway. People finally thought that they had found the key to the origin of life. This is sort of a schematic of the process that they were envisioning. In the first step here, this is what Miller did in his experiment. He took a simple mixture of gases and produced this complex mixture of fatty acids and amino acids, sugars and purines and pyrimidines. Then the idea was that these compounds would polymerize. The fatty acids would react with glycerin and make triglycerides, and these would be the basis for lipids. Amino acids would polymerize the peptides, and eventually the chains would be long enough to be called proteins and enzymes. The sugars, they would combine to make complex carbohydrates, things like starches and cellulose. And of course, the purines and pyrimidines, these are the building blocks of RNA and DNA. And then it was envisioned that by some process that they didn't quite know, all these things would get together and eventually make a true cell. And at first glance, this seems very reasonable. It's a very simple system. It's very elegant in its simplicity. You might even think it's so simple and so straightforward that it was designed, which is probably the worst thing you can say about a natural process. Now, at the time, George Wald wrote an article for Scientific American where he picked up on the Miller experiment and sort of elaborated it as a possible pathway, or the first step in the pathway to the origin of life. And I have some quotes from his article. And what he said was, however improbable we regard this event, or any of the steps which it involves, given enough time, it will almost certainly happen at least once. He was totally convinced that once you make the basic building blocks, things would all come together. He says that time is in fact the hero of the plot. The time which we have to deal is on the order of 2 billion years. Later in the article, he said, what we regard as impossible on the basis of human experience is meaningless here. In other words, forget everything you know. Given so much time, the impossible becomes the possible the possible probable, and the probable virtually certain. One only has to wait. Time itself performs the miracles. Which is a very curious thing for a scientist to say. He's probably just being very flamboyant here, but he's talking about miracles. And that gives you an idea of how complex this process really is, because it's something that we wouldn't normally see, we wouldn't normally envision happening. But he said time was enough. Once we had those basic building blocks, we only had to wait. Well, not everybody was convinced by his article. A few years later, about a decade, there's an anonymous editorial that appeared in Nature. And they took a totally different view of the situation. They said that those who work on the origin of life must necessarily make bricks without very much straw, which goes a long way to explain why this field of study is so often regarded with deep suspicion. Speculation is bound to be rife and has also frequently been wild. And what they're saying here is there's not very much data. That's the bricks without straw. So there's a lot of speculation, and people are speculating quite wildly. Further in the article, they said that some attempts to account for the origin of life on Earth, however ingenious, have shared much with imaginative literature and little with theoretical inference of the kind which can be confronted with observational evidence of some kind or another. In other words, it's all science fiction. It's not been tested in the laboratory. So in fact, when you go back and you look at this scheme for abiogenesis, this is the part that Stanley Maillard did. But nobody at the time, while writing his article, knew how any of these steps have happened. And today, it's still pretty much that case. There are a lot of question marks. You don't know how these things are happening. And in fact, the more you look at this figure, the more it looks like this cartoon, where you have a lot of stuff at the beginning and you have what you want at the end. But in the middle, you have a miracle happening. About 25 years after he wrote that article, Scientific American did a compendium of different articles on the origin of life. And when they reproduced or reprinted Wald's article, there was an editorial that preceded it that basically retracted what he said. And what they said in the editorial is that although stimulating, this article probably represents one of the very few times in his professional life when Wald has been wrong. Examine his main thesis here and see, can we really form a biological cell by waiting for chance combinations of organic compounds? Sir Fred Hoyle likened such an occurrence to the assemblage of a 747 by a tornado whirling through a junkyard. From what I've seen of tornadoes, this is not likely to happen. Later in the editorial, they said Harold Morowitz, in his book Energy Flow in Biology, computed that merely to create a bacterium, this would be one of the simplest organisms, would require more time than the universe might ever see if chance combinations of its molecules were the only driving force. Now they're not talking about the time that has preceded us, but he's talking about the whole lifetime in the universe would not be sufficient time for chance combinations. And that's a little hard to think about. So what I thought I would illustrate is how many chance combinations of amino acids there are. Now in Miller's experiment, he found about 7 or 8 amino acids, but by the end of the 70s, about 50 different amino acids had been found. And all but 2 have at least 2 optical isomers. And some of them have several centers of optical isomerism, so you have 2, 4, and 8 possibilities for those amino acids. And you need a minimum chain length of about 100 residues before a protein begins to have enzymatic activity. And for each amino acid, there are at least 2 ways of reacting. They have an amino group, they have a carboxylic acid group, they can form the peptide bond, but they could also turn around and form an acid anhydride. They could also react with side chains for the ones that have amino or acidic side chains. And the longer the chain gets, the more possibilities you have. But what I did for this calculation was just say, there's just 2 ways. We'll keep it simple. So the number of possible combinations, if you do the math, you've got the 50 amino acids, the 2 optical isomers, the 2 ways of combining, and you need 100 of them in a chain. So you work through the math, and you come up to a number that's 10 to the 230th power. If you're not familiar with scientific notation, that's a very big number. To give you an idea how big that number is, what if we just had 1 of each of these molecules? How much would that whole thing weigh? Well, to get the weight, we take the number of molecules times the average molecular weight of the protein and divide it by Avogadro's number. So the average molecular weight of an amino acid is about 130, 140. That's 100 times the average weight. And then when you put these amino acids together, you make 99 peptide bonds. So we have to take out 99 times 18, which is the weight of water that comes out from that peptide bond. And when you go through and do the math, the average weight of one of these 100 amino acid peptide proteins is 8,218 Daltons. To get how much they all weigh together, we take the 10 to the 230th power times the weight divided by Avogadro's number, and we get an answer that's 10 to the 210th power grams. That's a lot. To give you an idea how much a lot it is, the mass of the universe, the whole universe, not just the known universe, but everything, including the dark matter, is on the order of 10 to the 56 grams. So we would need in excess of 10 to the 150 power universes, that mass, to make all these amino acids. So we had just 1 of all the possible combinations. So random chance combinations of amino acids produces a huge number of possibilities. But only some of them are going to have enzymatic activity. And those are the ones that you want. You have to pick them out of this big mass. That's going to be a difficult problem. But what really killed Wald's thesis wasn't calculations like this, but it was the discovery of new evidence. And it was startling new evidence. Between the early 50s and the middle to late 70s, Barghorn and Schopp had discovered in rocks microfossils, fossils of what looked like bacteria and algae. And these were in rocks 3.5 billion years old. More recently, deposits with carbon isotopes characteristic of living organisms were found in formations 3.8 billion years old. The problem you have is that instead of having 4.5 billion years to 3.8 to make life, the Earth wasn't habitable for the first 700 million years. Depending on how fast it formed, there are different ideas, but in most of these, you have the Earth, its surface being too hot. You don't even have liquid water, it's so hot. Then you have another problem that around the 4 billion to 3.8 billion year time period, there was what's known as the late heavy bombardment. Looking at cratering on the moon, there was the second episode. After the planets and the moon and everything had formed, there was this second episode of very large meteor impacts. And it's thought that they may have been sufficient to melt the surface of the Earth, or if not, at least to make it uninhabitable by life. So you don't begin to look for life to form on the Earth until about 3.8 billion years ago. But we have evidence that life may have been existent almost instantaneously. If the carbon isotope work turns out to be invalid, we have visible algae and other microfossils in rocks at 3.5 billion years. So at most you have is 300 million years. You don't have the 2 billion years that Wald talked about. And I have no doubt that as more discoveries are made, this time window for life to originate is going to get smaller. So there's this exceptional little time for life to form by unguided chemical reactions. And that's why Wald's idea that time was sufficient to perform the miracles was dismissed. Currently, when people talk about the origin of life, what they're trying to do is to give us what they call a coherent narrative. They know that they're having problems identifying what the early Earth was like, but they want to give us this story, if you will, of how life originated. Now that's nice, but if we don't check that story against reality, it's just a story. And we're trying to do a scientific evaluation here, and stories just aren't going to be good enough. We need to be able to test these things. Now, if you want an idea of where things stand presently, there's a very nice book written by Jeff Beta and Christopher Willis, and it's called The Spark of Life, Darwin and the Primeval Soup. And in there, they've collected a lot of the current thought and a lot of the current work on the origin of life. But it's presented as a nice, coherent narrative. They don't do a scholarly criticism of this work. This is a popular book. They don't want people to get all wrapped up in controversy. They just want to lay out how it might have happened. On the other hand, there are people that, like the Nature editorial, aren't buying it. A friend of mine and a member of my thesis committee, Ken Nielsen, he said, "Nobody understands the origin of life. If they say they do, they're probably trying to fool you." When I found this quote from Ken, I thought, well, that sounds rather cynical. And maybe he didn't say it because I found it on the internet. So I wrote to Ken and I asked him, "Did you really say this?" And he said he did. And while I probably am not as cynical as Ken is about this, that they're probably trying to fool us, they may have managed to fool themselves. And that's something we all have to be careful about, especially in an area like this where there's so little that we can actually test our ideas against. So the question is, how do we get into this mess where we've got some scientists that say it's not a problem, it all works out, there are all these possible reactions, and in the end, all the compounds come together to form a cell. And on the other side of the fence, there are scientists saying, well, nobody knows what they're talking about. They're all just trying to fool you. How we got in this mess is actually a long story, but I'll try to be brief. The concept of spontaneous generation is a very old one. The Greeks considered this a possibility. Empedocles was first proposed that life arose naturally. He envisioned this as a continuous, ongoing process. It doesn't happen just once, it happens continually. And that the forms that persisted were those best adapted for survival and reproduction, which sounds very familiar to me. But we won't worry about that tonight. His ideas were repeated and expanded upon by Epicurus and Lucretius, other Greek philosophers. But Plato and Aristotle rejected this idea that life is constantly forming. They saw design and purpose in nature. And for them, design implied a designer. Now, jumping ahead a couple thousand years, we notice that Darwin, when he wrote his book Origin of Species, did not include any discussion of the origin of life. And that seems to me like a rather big hole given the title of his book. Did he simply duck the question? Or maybe he just thought, well, it's outside the scope of my discussion. Or did he consider the origin of life a given, since at the time the concept of spontaneous generation was still a viable idea? People still thought that life arose spontaneously. Well, what we know is that the Origin of Species was written in 1836 to 1844, wasn't published until 1859. But the experiments that Pasteur did to disprove spontaneous generation were not done until 1860 to 1864. So it's possible Darwin considered that the spontaneous origin of life was something very possible. What we do know is that in 1871, 7 years after Pasteur published his results disproving spontaneous generation, Darwin wrote a letter to his friend Joseph Hooker, where he said, "It is often said that all the conditions for the first production of a living organism are now present, which could ever have been present. But if, and oh what a big if, we could conceive of some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes. At the present day, such matter would be instantly absorbed, which would not have been the case before living creatures were formed." So he's envisioning, you got this warm little pond with just the right ingredients for making a cell, and they all just come together. Now, it was another 50 years before anybody produced anything of substance along this line. And what happened is 1924, Oparin, and in 1928, Haldane. Oparin is a Russian scientist, and Haldane is a British biologist. Separately and independently proposed that under reducing conditions on the early Earth, amino acids might be formed, and somehow this primordial prebiotic soup, life somehow emerges. So they envisioned a reducing atmosphere giving rise to Darwin's warm little pond. Well, it wasn't for another 25 years before somebody actually tried to test the idea, to actually see will this really work? And that's the now famous Maillard experiment, where they did indeed make amino acids using reducing conditions. Now, what they were trying to do in this experiment was simulate the prebiotic chemistry on the early Earth. And by prebiotic chemistry, we just mean the chemistry that happens before life begins. They took Oparin and Haldane's idea of a reducing atmosphere, and they applied an energy source to drive the organic compound synthesis. And they built this apparatus to simulate the early Earth. And their idea was you put the ingredients in, you turn on the electric spark, wait a few days, and in the end it would produce the building blocks of life. And they really expected to see just about every possible organic molecule come out of this. Now, what are reducing conditions? If we imagine that the foremost, well, we don't have to imagine, we know the four most important elements to life are hydrogen, carbon, nitrogen, and oxygen. In a reducing atmosphere, the hydrogen would be in the form of molecular hydrogen. It would be a gas. Carbon would be in the form of methane. Nitrogen would be in the form of ammonia. And oxygen would be in the form of water vapor. Our current oxidizing atmosphere, hydrogen is in the form mostly of water. There's some trace levels of hydrogen, but these are being continually produced, and they're rapidly consumed. Carbon is in the form of CO2. Nitrogen is the molecular nitrogen gas, and oxygen is molecular oxygen gas. So what Miller and Urey did is they used this reducing mixture. And this is the apparatus they designed. It has a large flask, a 5-liter flask, where they had their atmosphere. There were the 2 tungsten electrodes to make the spark that would simulate lightning. They had a condenser to simulate the cloud-forming processes in rain. They had a little trap here that would be filled with fresh water condensing from the large bulb. This would be like the freshwater streams running down the sides of the continents. And then they had an ocean here where they would be drawing their samples out, where the organic compounds should collect if they were formed. And they heated this flask so that the water vapor would go back up and circulate, and this would go around and around. And when they did the experiment, they really did find all the building blocks of life. They found amino acids, they found fatty acids, not real long chain ones. They found things like acetic and propionic. They found many of the nucleobases that you need for RNA and DNA, and they found some small sugars. But if you read Miller's papers very carefully, you'll also notice that he mentions this oily red goo coating the walls of his apparatus. It turns out this is the major carbon product of the experiment. But most people don't concern themselves with it because it's not one of the building blocks. These are what they were interested in, they focused on, and they just sort of ignored this for years. We'll come back to this later. Now, as I said earlier, the experiment was hailed as the key step leading to the natural origin of life. It was frequently imitated. There was actually a small cottage industry of maybe 25 or 30 scientists right after Maillard did his experiment trying to reproduce it. And they used a variety of different reducing atmospheres, a variety of energy sources, and the products they got were largely the same. There were some changes in distribution, but they all got amino acids, they all got fatty acids, they all got some of the purines and purinines. It seemed like this was a general process that happens whenever you have a reducing atmosphere. And they were all convinced that the first hurdle to forming life abiotically had been crossed. But the question is, did it really happen that way? Is there any evidence on the earth that that's how life actually originated? Or that we even had this time of prebiotic synthesis? And it turns out there's no rocks or sediments from this time of about 4 billion years ago. So there's no way we can test whether it actually happened. But there's also some rather serious problems with the Maillard experiment. The amino acids found were in very low abundance. Given the analytical techniques of the time, Stanley Maillard was lucky to find them. He used paper chromatography. His detection limits are thousands of times higher than what you can determine today with a gas chromatograph. Most of the amino acids that he found have turned out to be non-protein amino acids. These are amino acids that organisms do not use. They have different structures. And all the amino acids that he produced were racemic. In living organisms today, only the L form is used. And the reason for this is proteins, the peptide chain is a coil. And if you have a mixture of the D and L form, you can't form that coil. It kinks, it turns all different ways. What Maillard did not find was any peptides. He didn't get any homogeneous polymerization of the amino acids. And this homogeneous polymerization of amino acids to form peptides, it's thermodynamically unfavored, even when you have pure substances in solution. It's even worse when you mix the amino acids with other compounds. And most recently, the presence of a reducing atmosphere on the early Earth has been questioned. Some dismiss it as completely unlikely, such as Howard Holland at Harvard. And the problem we have there is we just don't have any real evidence of what the atmosphere was like on the early Earth. But the people haven't given up. Last year in Science, Chen et al. Had a new model for the atmosphere on the early Earth where they were able to produce a hydrogen-rich atmosphere. So this is a question that is still actively being debated today, and they'll fight forever because they don't have any samples to solve the question. We also don't know what the conditions on the early Earth were like. Was it freezing? Was the accretion of the Earth so slow that it was cold? Was it fast and we had a molten surface of the Earth? Or was it something in between? There just isn't evidence to resolve these questions. Now, shortly before I became a graduate student at Scripps, the Murchison meteorite, it fell in Australia. This was the year man landed on the moon. And although this meteorite landed way in the Australian outback, the people there, knowing that the United States was sending men to the moon, they thought this meteorite would be very interesting to them. So when they collected the meteorite, they kept it clean, they kept it uncontaminated, and they shipped it off to NASA Ames here in California and also to the Field Museum in Chicago, because they found several different pieces of it. And this meteorite was identified as a carbonaceous chondrite. These meteorites, the carbonaceous chondrites, are the oldest material in the solar system. And it's thought that they may contain some evidence that will help us to decide about life's origin. And shortly after it was sent to NASA Ames, the team that was supposed to look at the moon rocks for organic compounds got this meteorite and they analyzed it. It's a case of the prepared, chance favoring the prepared mind. They were ready to look at really low levels, and a sample just walked in their door one day. And what they found were very low abundances of amino acids. We're talking a few micrograms per gram of meteorite, so a couple of parts per million. And what they found in this pattern, found in the amino acids, was a pattern very similar to the Maillard experiment. Mostly, the amino acids were non-protein amino acids, which is also very similar to the Maillard experiment. And all the amino acids were racemic. So if you had an optical center, you've got both the D and L forms. But the most abundant form of carbon in the Murchison meteorite was a dark red to black tar. Most people don't pay much attention to that because it's not exciting like amino acids. So at the time that this discovery was announced, Stanley Miller and Jeff Beta, they came up with an idea. And they said, well, the Strecker cyanohydrin pathway was postulated as the way amino acids were formed in the Maillard experiment. But they noticed that this pathway is forked. The normal path, when there's lots of ammonia present, you get only amino acids. But there's an alternate path, and if there's no ammonia present, the pathway produces hydroxy acids. So what they suggested is if the ammonia concentration on the meteorite parent body was some moderate amount of ammonia, then both products, the amino acids and the hydroxy acids, would be formed, and they could use the ratio of the appropriate pairs to tell us about the ammonia concentration on the meteorite parent body. All they needed was two things. One was a sample of the Murchison meteorite, and the other was a graduate student that they could get to do the work. And I remember distinctly the day Jeff proposed this in class, and he baited the hook, and I bit hard. It seemed like a really exciting project to work on, and being a first-year graduate student, needing a thesis project, it was all just laid out there for me to take. And so I went and I talked to him, and he agreed to let me work on the project. What I didn't know is going to take me 3 years to develop the analytical method. The hydroxy acids are a lot more difficult to work with than amino acids. They're actually quite volatile. And so it was a painstaking process to develop the method. But I eventually got it to work, and I was all excited. I went to Jeff and I said, well, I'm ready to go. Let's get the sample of the meteorite and do the analysis. And he said, well, we better call Stanley Miller first and talk to him. And Miller wisely suggested that I test the method on one of the experiments that he had run, some of that extract from the flask, and see if I could find hydroxy acids in there. It should be about the same degree of difficulty. And then they could really test to see if my method did work. Wasn't that they didn't trust me, but there aren't many Murchison meteorites in the world, and you don't want to blow your one good sample. The method worked very well in the Maillard experiment. I got to do the meteorite, and the assay for hydroxy acids in the Murchison meteorite worked very well. We found an abundance pattern identical to what I found in the Maillard experiment. The hydroxy acids were all racemic, so I knew they were non-biologic origin. They weren't contamination from my fingerprints and things like that. And we were able to use the appropriate pairs of amino and hydroxy acids to calculate the ammonia concentration on the meteorite parent body. And it came out to 1 or 2 millimolar, which doesn't mean a lot to you folks, but it was very close to a number that Jeff had calculated on his thesis research about what the concentration of ammonia should be on the Earth due to the stability constants of some of the amino acids. So we're all very pleased and happy. This was great, exciting work. But it didn't generate anywhere near the fanfare that either the Maillard experiment did or the discovery of amino acids in a meteorite. Nobody knows what hydroxy acids are, and they really don't care. So I wisely decided that, well, the reason I was at Scripps was to get a PhD in work in oceanography, so I would pursue my career as an oceanographer and not worry about analyzing meteorites for the rest of my life. And that turns out to be a good decision because carbonaceous chondrites don't fall that often, about once every 30 years. And it's hard to build a career when you have 30 years between samples. So everything seemed to be well at when I finished my degree. But over the years, I began to think about some of the things and some of the implications of this work. Now, the Miller beta prediction that we would find hydroxy acids in the meteorite, that was confirmed. And this Strecker cyanohydrin pathway has been well identified as a source of the amino acids in the meteorite and also as a possible pathway on the early Earth. Or is it? The question of the reducing atmosphere is really key to this. If you don't have a reducing atmosphere, this pathway doesn't happen. Now, at the time that this reducing atmosphere became disputed, Miller said, well, not to worry, we've got meteorites now that make these, we find these amino acids and hydroxy acids in meteorites. Maybe the meteorites bring them to the Earth, and we don't have to have a reducing atmosphere. So he did the calculation based on estimates of the flux of meteorites and the concentration of amino acids in the Murchison meteorite. And he was very disappointed because it turns out that if you take all the amino acids and all the meteorites and put them in the Earth's ocean, you only get about 10 billionths of amino acids, 10 billionths of a gram per liter. And this is a very small concentration. Nothing's going to happen at those low concentrations. But people said not to worry. The world's oceans are actually, that water was brought here by comets, and comets are going to be loaded with organic compounds, and we just have to analyze the comets and everything will be fine. Well, we're still waiting for the comet analysis, so we haven't tested that idea yet. But if we look back at the Miller-Urey experiment, there's another problem. Why are the results from the Murchison meteorite and the Miller-Urey experiment so similar? The meteorite, it's 4.5 billion years old. It represents untold years of chemical synthesis. Maybe millions, maybe billions, we don't know. The experiment that Stanley Miller did was only 6 or 7 days. Why should the two look so very familiar? And the other question that lingered, the one that I've struggled with, is what is the source of this red goo in the electric discharge experiment and the black tar in the Murchison meteorite? These are the major forms of carbon in both cases. Where are they coming from? In economics, people say if you want to figure out what's going on, you follow the money, or in politics for that matter. Well, in chemistry, if you want to figure out what's going on, particularly with origin of life, you've got to follow the carbon. So we need to look at where are the major products, this red goo and the black tar, where are they coming from? So I looked at Miller, some of his old data from one of his early papers. And this shows the kinetics of the reaction. And you can see as time goes by, the ammonia concentration, it drops pretty linearly. And shortly after the experiment starts, hydrogen cyanide and aldehydes are produced, but they quickly plateau. They reach the steady state concentration. And a short while after, the amino acids start to come in, but they plateau as well. And these plateaus here, these are what chemists call steady states. And what's happening is this experiment really involves 3 steps. The first step is the synthesis of the aldehydes and hydrogen cyanide from the gases. These aldehydes and hydrogen cyanide then react through the Strecker cyanohydrin pathway to make amino acids, hydroxy acids. The third step is that something is consuming the amino acids. Because if you've got one process producing them, you have another one consuming them at balanced rates to get that steady state concentration. But there's no evidence of polymerization of the amino acids to peptides. We don't find peptides in this experiment. So the question is, are the amino acids reacting with other compounds and forming something else? And the answer is yes. It's called the Maillard reaction, and it's actually familiar to all of us. Here's Peppermint Patty, and she's working on her science project, and she had some trouble, but she actually came up with something. She made toast. Now, it's a rather simple science project, but she was very proud of it. And it's not the baking or the making of toast, but rather it's the baking of the bread for the toast that the Maillard reaction is very important. It was first described by Louis Camille Maillard in 1912. And what happens is the amino acids from protein, they interact with reducing sugars like glucose or lactose. And this is why you add milk when you bake bread and not table sugar, which is not a reducing sugar. And these compounds react to produce colors, aromas, and flavors characteristic of a particularly cooked food. Now, temperature accelerates the Maillard reaction. Products give bread crusts, cakes, and cheese pizzas their distinctive brown color. And the small volatile compounds that are produced as a byproduct of this reaction are responsible for the caramel-like and roasted aromas that you get when you bake bread and bake pizza. Now, for the chemists, I threw in this slide. The rest of you don't panic. It's not as bad as it looks. But here's a reducing sugar reacting with an amino acid. And it's the amino group that attacks the reducing part of the sugar through the Maillard reaction, producing amidori compounds. And these then undergo a whole cascade of different reactions giving different products. One of these is a Strecker degradation, where you lose CO2, amines, and you get back the aldehydes that you started with when you were making amino acids. All of these products then combined to produce melanoids. Now, melanoids is this large class of compounds. They have one thing in common, and that's they're all colored. That's why they're called melanoids. It's sort of one of those trash can terms that you can throw a lot of things into. You don't know what's in there, but they're all colored. I looked up the Maillard reaction on the internet a few years ago to see how widely it was studied. And it turns out it's a big deal. This was the 7th International Symposium. It was held in Tokyo and a few other cities in Japan. What was interesting is the two groups of people that were coming. You can't read the sessions here too well, but there are two kinds. One is medical science, and the other is food science. I would imagine this would be a great conference to go to, because all the food scientists, they're going to be interested in good food. So the meals there are going to be fabulous. And if you overindulge, there's all these medical doctors there that'll take care of you. As I said earlier, temperature accelerates the Maillard reaction. That's why we heat food to cook it. But it can also proceed at room temperature. And I think you've all seen this happen as well. If you slice an apple and just let it sit, in a few hours, it turns brown. And what's happening there is that when you cut the apple, you destroyed cells and released a lot of compounds. They're now free in that apple juice that you created to react on their own. They're no longer bound up. And they produce these colored compounds. During that same time frame, if you had a mixture of amino acids, you're not going to make any peptides. And if you cook something too long, you get melanoids and black tars, burnt toast. This reaction is also very important in aging. Proteins and carbohydrates react in a similar chemical manner at body temperature. You don't have to heat things really hot. The lens crystalline protein, that turns brown as people get old. This is part of cataract formation, is you're forming these brown colored compounds via the Maillard reaction in your eyes. End products of the Maillard reaction have been isolated from Alzheimer's disease lesions. They've also been implicated in arteriosclerosis and diabetes mellitus. In other words, we are slowly cooking as we age, and as a consequence of disease. I know it's not a pleasant thought, but it's one of the things that happens to us. The real question is, why don't we cook faster? The apple, after we sliced it, it only took an hour to turn brown. Our bodies at a much higher temperature, why isn't this reaction just stopping our biology cold? And the answer is, it's due to the cell structure. Amino acids and sugars inside a cell, they're not floating free. There are all kinds of internal organizations within a cell that contain the various substances. We have ribosomes to control protein synthesis, and there's the mitochondria to facilitate the energy reactions. And it's these structures that are keeping the sugars and the amino acids apart. So what we're seeing is the complex structure of the cell itself is needed to make the components necessary for making the cell and keeping them from reacting uncontrollably or randomly. So how does this all relate back to abiogenesis? Well, we don't know how any of these reactions could possibly have occurred on the early Earth. But we do know that if you've got a mixture that's composed of this, it's going to react and produce melanoids and eventually, with enough cross-linking, go to kerogen. This is the red oil that Miller saw in his experiment. And this is the black tar that we find in the meteorite. So we know from what observations we made that the second step in the Miller-Urey experiment was not the production of biopolymers, but were these colored, what we call geopolymers. This means that the Maillard reaction is really a showstopper in terms of abiogenesis. And it's not just in his experiment and in the meteorite that this reaction is important. If one studies organic geochemistry, you'll probably see this picture somewhere in your class. And what happens when organisms die, their cells break apart, they release the carbohydrates and the lipids. All these biopolymers are released into the environment. Through microbial degradation and also inorganic decomposition, these biopolymers are broken down into their component parts: sugars, organic acids, amino acids, fatty acids. This mixture should be very familiar to us by now. This is what Miller produced. In the natural environment today, these compounds are reacting through the Maillard reaction, producing complex assemblages. They're polymerizing. We've got fulvic acids being produced, humic acids, and as they continue to cross-link, they produce the geopolymers, the melanoids and the kerogen that we find in rocks and sediments. So the difference between biopolymers and geopolymers, structure-wise, biopolymers are linear. Proteins are a linear chain of amino acids. In the geopolymers, things are branched. They go every which way, and we have all kinds of bond types. Here we have just the peptide bond. The components, well, in proteins, we have amino acids. In DNA and RNA, we have the nucleic acids. In starches and cellulose, we just have sugars. So it's just a single compound class. In the geopolymers, they're all mixed up together. Functionally, the biopolymers act as catalysis. They sort of a form of energy storage in the starches, and they make up our genome. The geopolymers, it's tars. They have no chemical functions. In terms of information content, there's all kinds of stuff stored in our genome in the DNA and RNA. There's no information in the geopolymers. So what does all this apply for the Miller-Urey experiment? Well, the amino acids that we found are intermediates in the reaction pathway, not the end products. And what we find are actually remnants of the reaction. And that's why you find the low yields. The prebiotic synthesis makes melanoids. This is the oily red goo that you saw in the flask from the building blocks of life. Prebiotic chemistry is essentially over as soon as it starts. You don't make the biopolymers that you need, no matter how much time you have, because you're making the melanoids. Compare this to what you find in a single-cell organism. Single-cell organisms like Prochlorococcus, which is a marine photosynthetic prokaryote, in a single day, it can make all the chemicals it needs to make an exact copy of itself. This single cell is, it's capable of all the complex biochemistry that's essential for life. And cellular structure is essential to the process. Without this cellular structure, as we saw with the apple when you slice it, the Maillard reaction takes over and makes melanoids. So with this structure, it's possible to make the homogeneous polymers that we need. The cell function grows and reproduces itself. We're now at a point where we can say that, well, the cell is an irreducibly complex structure. You can't make it piecemeal. Only cells can make cells. This was something that Pasteur was proving in his early experiments. Life was coming from life. It wasn't forming spontaneously. So we've got this cell that the only way we can make it is the processes in that cell making a copy of itself. So a quick summary so far. Despite the apparent success of the Maillard experiment in 1953, many problems remain. Re-examination of this experiment shows that this pathway is not the road to Darwin's warm little pond, nor is it a pathway to biomolecules or microbes. Think of what happened with that apple. We're going to be making melanoids, not microbes. On the other hand, complex cellular structures are essential. They are an essential characteristic of and a prerequisite to life. You've got to have these things. So the origin of life requires a level of complexity that undirected chemistry cannot achieve regardless of how much time is available. We've got this chicken or egg situation. We need the cell to make the compounds that are in it, and we need the compounds that are in it to make the cell. Which came first? Biologists recognize how difficult this problem now is. F.M. Harold, in an article in 1995, says the origin of life stands as the most profound mystery in biology. And we note in passing that despite all the achievements of investigators in prebiotic biochemistry, it remains utterly beyond our comprehension. That's a fancy way of saying what Ken Nielsen said earlier. Nobody knows how to do this. Lynn Margulis, who was one of the main proponents of the abiogenesis, she admits that to go from a bacterium to people is less of a step than to go from a mixture of amino acids to a bacterium. She's recognizing that this first step, the first living organism, is the most difficult problem. Because of these obvious problems, I think if we're honest, we will recognize that abiogenesis is at best, it's a myth of modern science. It's a nice coherent story that we tell ourselves. But at worst, it's a lie we tell ourselves so we can pretend we know more about the origin of life than we actually do. We are actually fooling ourselves when we say that this is the process that life began. So what's a chemist to do about this? Well, they've tried lowering the bar. Let's not try and make the whole cell. Let's try and make just different parts. Well, synthesizing the building blocks, that's done. But now we've got to separate the building blocks to keep them from reacting. Chemists can do this acting intentionally, but how does this happen in nature? What kind of random process pulls a mixture apart? Another simple step would be to make some basic biopolymers. But how does that happen in nature when, if you put the polymers themselves in water, they hydrolyze and come apart? Then the idea is, well, once we've got the basic things started, some sort of chemical evolution will occur and we'll evolve more complex molecules. Now, I have to say, honestly, now that I'm not a graduate student, thinking about this, it's probably the most crazy thing I ever heard. Molecules don't evolve. They're chemicals. They react, and they become another chemical. But to evolve means that they have some sort of reproduction. They don't. And the last step was assemble a minimal cell. If we can't make a complex cell, let's try and make something very basic. So these are all ideas that people are trying to use today to advance the idea of abiogenesis. One of the big ideas that's being pushed is these self-replicating molecules. Another idea is that proteins came first. And the idea is once you have some proteins, maybe they helped make the DNA. Maybe proteins and nucleotides appeared together and they worked and helped each other out. Maybe metabolism came first. Maybe membranes came first. And there are other ideas. But let's just look at these first 5. These are ideas of breaking the process down into smaller steps. Leslie Orgel is one of the big promoters of self-replicating molecules. And this is an idea that he has proposed. Now, he hasn't done the experiment. This is just an idea that needs to be tested. And you have this molecule down here that, because of its various functional groups, attracts these other 2 molecules together. And when these 2 groups react, and these 2 groups should be very familiar, because here's the amino group and the aldehyde, and this combination here is the first step in the Maillard reaction. They join together, and now you have a molecule that's identical to the one you started with. Now, the curious thing here is, this looks like a great idea. But remember, these are the 2 functional groups that make the Maillard reaction. So this group can react with any aldehyde. So if it's not bound up here and it's floating out, there's other competitive reactions that are going to be going after it as well. And the same is true for this aldehyde function. Orgel also very cleverly used the same names for the side chains, R1 and R1 and R2 and R2, so that his condensed molecule would be identical to the starting one. But there's absolutely no way that this molecule controls what these side chains are. These could be anything. So it's not really a self-replicating molecule. It is actually just a template that might help other molecules come together. What he's done here is a semantic illusion. I don't know whether he's intentionally trying to trick us or whether he's managed to trick himself as well. But this isn't a self-replicating molecule. The idea of the proteins first and proteins and nucleotides together, Manfred Eigen is one of the big proponents of this. His thoughts are that enzymatic activity is first can aid the synthesis of the genetic material. Well, we have these problems. There's no evidence that proteins form naturally. And in the experiments that he's done to get these things to happen, he's done what has been called investigator intervention. This is things like eliminating all the competing reactions. So you only put amino acids in when you're trying to do a polymerization. It's things like adding leaving groups to the ends of the molecules so they join together and the reaction actually happens. And it's other things like putting blocking groups on the molecules where you don't want them to react so they join together the right way and not the random way, which is what would be happening in nature if he wasn't there. And he's worked with Freeman Dyson on this double origin of both nucleic acids and proteins. And the idea is there's this synergy between the 2 compound classes. But if we think about it, without the protection of a cell wall, both materials will be consumed by the Maillard reaction. So these are nice ideas, but when you really put them in nature, there are the competing reactions that are very fast that are going to outcompete. To help catalyze these processes, Karen Smith has said, well, maybe clays are somehow involved. These organic compounds are absorbed on the surface of clays, and that gets the thing going. Well, just a week or two ago, David Deamer, there was an article in Science about David Deamer from UCSC. He added a mixture of proteins, DNA, and cell membranes to a hot little puddle in Kamchatka, Russia. And when I first looked at this picture, which was a little hard to see, here he is pouring a beaker of some liquid. I thought, well, is he just pouring a quart of milk in? But reading the news article with it, they actually had this mixture of, that he made up of proteins, DNA, and cell membranes. Well, where did they come from? I mean, shouldn't he be starting with amino acids, nucleotides, and fatty acids? I mean, he's a couple steps ahead already. Well, what happened as an experiment was that the compounds he added, within a few hours, were totally absorbed by the clays, and that's where they stayed. They were completely taken out of solution, and they were done. Nothing happened besides the clays absorbing the organic compounds. So clays don't actually help catalyze these processes. What the clays do is they strip things from solution, and the prebiotic soup just gets more dilute with time. Harold Morowitz is a big proponent of the reverse tricarboxylic acid cycle. This is the cycle that cells use to generate energy. And he uses this to explain the production of long-chain fatty acids that can be used to make cell walls. Well, this process involves the directed input of hydrogen and carbon dioxide. And there's no way to contain the product. So even if it works a couple times around, as you start to build up a longer and longer chain, it's going to drift away. You need a cell to keep the products there. Also, many of the products of this process are subject to degradation by the Maillard reaction. That reaction is just killing everything. Because they're fatty acids, they have the ketones and aldehyde functions that can be attacked by amines. The other process is naked undirected chemistry, chemistry outside a cell. It's not cyclic. Chemicals react and they go downhill thermodynamically, and they go downhill very fast, but they don't go around in cycles. Another idea is that, well, maybe you have membranes first, and you get things called these protocells, or coacervates, as they used to be called. And they capture chemistry and take it inside. Well, that's a nice idea, but as soon as the chemistry inside the cell reacts, it's done. You'd have to worry about diffusion through these cell membranes to get more supplies in to keep the thing going. And if the cell membrane was porous enough for things to come in, well, all kinds of things would come in, and you'd pretty soon have the Maillard reaction going on inside the cell. So what is happening in all these examples is people are just grasping at straws. Many of these problems that they had before still remain. Biopolymers are not formed by random reactions. The various compound classes must be separated, or they're going to react through the Maillard reaction and consume each other. The biopolymers have to be assembled in very specific ways. Proteins require a peptide bond. They're just not any old random linkage. The biopolymers are composed of hundreds of monomers arranged in a specific order. There's information content in these molecules. Like I showed earlier, if you just do it randomly, there's a vast number of combinations, but only a few of them are ones that actually work. And cells require hundreds to a few thousands of enzymes in specific places. The genetic material must be isolated, and the energy compounds must be utilized properly. This is a complicated structure. It's just not a bag of loose chemicals. Bruce Alberts, current and former head of the National Academy of Science, and a well-known biologist, he says that the cell is a biochemical assembly line. It's a little factory. All the factories I know have been designed. So what we need to do is we need to recognize that what prebiotic chemistry produces, the melanoids and the geopolymers, is really of no use to the origin of life. And what we need, the biopolymers of specific compositions, prebiotic chemistry cannot produce. Well, results so far indicate that life is not the result of unguided chemistry and random processes. It just hasn't happened. Life is also not the result of purposeful physical laws and initial conditions. We've put these chemicals together and they're just not doing what we need them to do. The laws of nature and fine-tuning of the initial conditions, we need to recognize that they are insufficient to account for the origin of life. Limiting our options to just natural processes alone, in fact, inhibits the origin of life. You're just not going to get there with what natural random reactions do. Well, if nature is insufficient, then maybe it's time to consider other options. One of the ideas, as crazy as it sounds, that's actually becoming very popular today, is that life came here from outer space. It was first proposed by Svante Arrhenius. And he proposed that bacteria, being pushed by light beams, came from other planets to the Earth. This idea was recently championed by Fred Hoyle and Wickramasinghe, and also by Francis Crick. Now the problem with it is that's how life came to Earth. It may explain why there's life on the Earth, but it doesn't tell us where life came from, because it puts the origin of life out there somewhere on another planet where we can't even begin to look at the conditions. So I find it a very unsatisfactory explanation. And there's all kinds of problems with getting things here through space, which I don't have time to go into. But if we're so desperate that scientists are seriously considering panspermia, maybe it's time for us to consider other options as well. Maybe it's time to consider design. If the cell is a miniature biochemical assembly line, then maybe it was designed by a prebiotic intelligence. Directed chemistry can make all the biopolymers we need, but we don't know which to make a priori. As a graduate student, I was able to separate all the compound classes to do the analysis. Today, graduate students, they can make enzymes, full, working, functional enzymes. People are trying to assemble viruses from scratch. We're at the point where we can make the biopolymers that are necessary. The trick is, people are just copying what's already been done. We're not randomly putting these things together. We're putting them together, matching patterns that we already find in nature. So we're not smart enough to know how to put them together. And once we've got the compounds, we're not smart enough how to know to put them back into a cell. When people do tissue experiments and they extract them or put them in a tissue miser and they break up all the cells and they want to pull out the different components, nobody ever worries that they're going to spontaneously go back together. This isn't something that just happens. The cells have to be carefully assembled. And we just don't know how to do that. So if cells are done by an intelligence, it's an intelligence smarter than we are. And clearly, that's what's needed. I'll conclude with two thoughts. One is from Karl Popper. He said this over 30 years ago. It's even more true today. And that's the undreamt of breakthroughs in molecular biology has made the problem of the origin of life a greater riddle than it was before. We have acquired new and deeper problems. What Miller was trying to do 50 years ago has now gotten harder. The complexity of cell is way beyond what we knew then. The other advice that I'll leave you with was written by Saul of Tarsus in the 1st century. Test everything, hold on to the good. I've thrown out a lot of ideas here tonight. I encourage you to, or challenge you to test them. Hold on to the good ones, throw out the bad ones, but don't just take my word for it. Do some investigating for yourself. And thank you very much.
I'm sure some questions would be answered if anyone has anything they'd like to put forward. If you do so, please come to the microphone.
Mike, while they're pondering, I'll put up a suggested reading list for people that want to investigate some more. These are 5 books that they might find of interest.
You mentioned it a number of times, but you didn't really go into it, the genetic code. I mean, of all the things that you point out, it seems the most wondrous is that we can have the level of information that we have in the genetic code put together in a certain sequence, not only of its own right, but the fact that it can also then template all protein and all other manufacturing that takes place in the cell, in the DNA. I mean, the amount of information content in there is larger than maybe in all the books in all the libraries in the world in one particular cell, in that one string of DNA, 10 to the 10 bases. And I wondered if you'd like to comment about that.
Well, there's the information content is really overwhelming, that it's in a really single molecule. The other thing about the genetic code that is very curious is there is no chemical reason for why the base pairs code for different amino acids. This is a code that could have been written anyway. The code does code for specific amino acids now, but it doesn't have to be the way it is. Because of the way it's transcribed, the RNA molecules, they pick a particular amino acid, but that could be assembled any way. This is a code that is purely arbitrary in development. And those kind of codes speak to me of intelligence. There is no underlying physical reason why a certain triplet of base pairs codes for a particular amino acid. This is something that was assigned early on. And whenever I see a code, well, I think of a code writer.
Yeah, one thing I was very interested in is if you had ever come across any attempts to explain the fact that all of the amino acids in proteins are left-handed instead of right-handed by naturalistic processes.
Okay, that's a very good question. It's one that there are no satisfactory answers for. You'll see it in lots of places where people are saying, we still don't know why. It's just L amino acids. In the book by Willis and Beta, they talk about a scientist that did an experiment where he built an enzyme using all D amino acids and found that it worked on the mirror image substrate that the L protein did. So there is no a priori chemical reason why it's all L or all D. It has to be, but it has to be one or the other. Because if you want to build a coil of amino acids, they all have to go in the same direction. Likewise, with DNA and RNA, you use D sugars. And the reason for that is with the D sugars, the 5 carbon is above the plane of the sugar, and the 3 hydroxy group is below. So you can build your phosphate bridges this way and have the base pairs come off. If you use L sugars, it goes the other way. So you could go in the other direction. But if you mix them up, you can't build that nice chain of nucleotides and make the helix.
Thanks.
I have one more question. I got the impression from the summary at the end of your talk that you don't hold out much hope. I mean, that there's avenues, most of the common avenues that could possibly have been in place on the early Earth, in the pre-cellular Earth, have already been explored, and they haven't led anywhere. I mean, so is this field going anywhere? Are there people that are still actively pursuing things, or are we at sort of the point of view of just postulating, well, we don't know how it happened, it just happened, and we're going to leave it there state? I mean, where is this field?
Well, is the field going anywhere? Are there people still active? Two questions. Yes, it's still active. There are still people trying to do these kind of experiments. I'm not impressed by their results because frequently, if you look at them carefully, there's a lot of investigator intervention. You know, they're stacking the deck for things to happen. And they're doing things that are not conceivable on the early Earth. You don't find amino acids with leaving groups and blocking groups so that they'll form proteins the way you want them to. So yes, there's people still active, but I don't think it's going anywhere. I think it's run its course. I think they've missed a lot by not paying attention to the fact that in the Maillard-Urey experiment, the walls were coated with polymers that were forming as fast as the amino acid were being produced. They've missed an essential part of the chemistry of the early Earth. If you're going to have a synthesis in a reducing atmosphere, you're going to produce all these compounds in a mixture, and it's going to produce this red, oily goo. And that's going to be its major product. And what it does is it takes the vast majority of the compounds that you produce, and it makes the red oil. Eventually it becomes a tar. That's not going to leave much left to build things with. It's been 50 years since the Maillard-Urey experiment. It's still the premier experiment. And I think it's time for people to look at this in the harsh light of day and say, we're not getting anywhere. I think back to the problem with alchemy, where 500 years BC, it was proposed that there were 4 elements: earth, air, fire, and water. And these were, everything was made from these 4 substances. And so you could take lead and make gold if you just took out the right amount of earth and put in enough fire or whatever. 2,000 years went by of failed experiments trying to do this. And finally somebody said, well, you know, maybe lead and gold are elements themselves, and we can't make one from the other. And it was that idea that quickly led to atomic theory and the modern periodic table. That never would have happened if they just stuck with the 4 elements. And I think what's happening with the abiotic chemistry research, the prebiotic chemistry research, is they're still on that alchemical stage where they're looking for the magic combinations of the 4 gases or some other equivalent of that. And they're not thinking outside the box that more is needed than just natural reactions. They need information, and they need to direct that synthesis. And those aren't natural processes, but that's what's needed to get a cell.
Does chemistry have anything to do with the specified complexity in the DNA?
No. The complexity of DNA, that's a code that's written in. You could, by changing the RNA that reads the code and is transferred to where the proteins are made, they carry an amino acid with them. You could take the part that reads the code and the part of the molecule that carries the amino acid, and you could put them together any way you wanted to. And you could change that code. It'd be a lot of work, but there's no reason why a certain triplet codes for a certain amino acid, other than the fact that it does, and because of that, it codes for the RNA that carries that transfer. It's information that's built into the code, but it's not required by any chemistry.
So chemistry couldn't really produce the information?
No. No. And like the amino acids, the number of combinations of nucleotides that you could come up with is enormous.
It seems you alluded to the problem, and it may be well understood. I'm just not that familiar with it. But one of the critical things is that you also need an RNA to transcribe the DNA if you ever got something like your 100-molecule chain. Is there any accepted model for how the RNA would have developed to transcribe that information and actually give you a replicating system if you even got the necessary complexity in the first molecule?
The simple answer is no. One of the things that Orgel is promoting with the RNA world is that RNA is both the memory compound and the transcriber. That way he's lowered the bar. The first organism is just a pure RNA organism. And it has RNA doing the memory work, it has RNA doing the transcribing work, and he may even have some RNA doing some enzymatic work. This seems to him a simpler task to achieve. But it creates a whole new set of problems, because how do we get from that RNA world to our current combined DNA RNA world? That's just as big a problem as creating one or the other.
And wasn't that the slide in which you pointed out that he had no control over the side chains?
Right.
But there's another question that interests me, that even if you assume that somehow that came to exist, is there any, if you think of the problem of assembling such a molecule the first time, is there any kind of estimate of the time that it would take to form such molecules, or are those of reasonable simplicity where that they could fit into that sort of 300 million year window?
The only estimate I know of the time involved was Harold Morowitz one, where he said the whole time of the universe is greater than what we need to get these things happening by random reaction. It's a big problem, and mostly people say, well, we got lucky. We got really, really lucky if it happened by random chance. It makes the lottery look like a sure thing.
Good.
Intelligent design may be something that the philosophers of science are able to develop a coherent research program around, but do you think that intelligent design are you proposing that become part of science, or something that simply gives perspective to the science that we do? In other words, it might be a useful religious or worldview or philosophical idea that makes sense of the world. The science we do describes the world that we live in, but it cannot answer certain questions. How do you think intelligent design and science work together? Or don't they?
Well, I think it's still early to say exactly how it's all going to come together. I think there are some research programs that intelligent design will push forward. There will always be the charge that it's religiously based. But limiting ourselves just to natural reactions or natural processes and saying that nature is all there is philosophically based. Currently, science has been abducted, if you will, by naturalists. And they've closed the doors to thinking outside their box. It's causing some problems, specifically looking at this question of the origin of life. It's pretty clear. Natural reactions aren't capable of doing what we need to be done, and what they do is not what we want. And so we need to get outside that box. One way to do that is to say that there is more, or there was more, operating at the origin of life than just the natural reactions. And we can see how design processes can accomplish many of the tasks. What you may not be able to say is, well, did this really happen this way? We have to go, we have the problem, we can't go back to the past and look at it, and we don't have much evidence. But it does present a process that is capable of accomplishing the task. The other issue with intelligent design is it doesn't specify the designer. But many of the opponents of intelligent design insist that it's being presented as a way of sneaking the conservative Christian God in the back door of science class. Let's turn that around. If we were insisting that intelligent design pointed to the conservative Christian God, we would never be able to defend that position because there's so little about the designers that revealed in this process that it'd be hard to make that conclusion and justify it. But it's one that's been hung on the ID movement by the other side. I think the resolution of the problem is to say there is evidence, or how does one decide about evidence for design? We need to look at that question and then ask, okay, now that we know how to identify design, do we find evidence for it in nature, in biology? And that's a two-step process. We're just at the beginning of the process of identifying design. And that's mostly a mathematical process at this point. But as that develops, it will then allow asking the second question. And those are valid questions that can be asked and studied scientifically by experimentation. I think that's where it may help us. Well, thank you all for coming, and thanks to the Veritas Forum for inviting me. It's been a real pleasure for me, and thanks for sticking out. I know it went a little long.
Thank you very much.